All right, well, good afternoon, everyone. I am Alex Mendenhall, a faculty member here in the department, and it is my distinct pleasure to introduce to you Dr. Xinxian Deng. And he was educated in Fudan University for his undergraduate. He got his PhD at Tufts, and then he did a postdoc with Dr. Christine Dosage here at the University of Washington. And now he is his own independent research assistant professor and will soon be coming up for a research associate professor. And he studies the basic biology of X-linked gene regulation and how that relates to human health and disease. So I'm sure we're all excited to hear what he has to say today. So with that, I will give it up to Dr. Deng.
Can you guys hear me now?
You sound great.
Okay. So first, thank Alex and the nice introductions, and also thank Alex and Scott for the opportunity to present our work on the X chromosome regulations for the past seminar. It has been a while since my last time talk for the past seminars. So sounds very familiar with this pathology seminar. So now let's talk about some sex chromosomes, which are the major object we're working on in the lab. In diploid organisms with a pair of chromosomes used for the sex determinations, males have one X chromosome and two Y chromosomes, while females have two X chromosomes. There is a dosage imbalance between the single X chromosomes and the two sets of autosomes in males, as well as an imbalance of X-linked genes between males and females. This imbalance is could cause lethal because due to the monosomy of the hundreds of genes on the single X chromosomes in males. Different species using the different mechanisms to adjust this imbalance to avoid the lethalities. In mammals, Drosophila C. elegans, males will upregulate genes on their single X chromosomes by up to twofold to achieve similar levels as two sets of the autosomes. To avoid overexpression in females with two X chromosomes, different species using different strategies. In mammals, female will inactivate one of their two X chromosomes, while in Drosophila, the X upregulation system is inhibited. In C. elegans, hermaphrodites will repress both of their two X chromosomes. Thus, dosage compensation of the X chromosomes involved in a balance between X upregulation and repression. So today I will talk to you about our work related with three major questions in the dosage compensation of dosage regulation of the X chromosomes in mammals. The first one is, how is the active X chromosome upregulated? Second is, how is the inactive X chromosome regulated? And what are the impacts for the X chromosome regulations on the human development and disease for the third question. So first about the X upregulations. We and others have already found the diverse mechanisms involved in the X upregulation in mammals. As a model presented here. Originally, the proto-X chromosome and the proto-Y chromosome have a very similar chromosome length as they derived from a pair of ordinary autosomes. So they will produce similar expression products compared to two sets of the autosomes. During evolutions, proto-Y will subject to the gene loss segment by segment due to the inhibition of the homologous recombination between proto-X and the proto-Y. Then for genes retained on the proto-X chromosome, they become hemizygotes. And if they are dosage sensitive, they will subject to the dosage regulations. So there are at least two types of the feed-forward regulations involved in the X upregulation. One type is transcriptional regulations, including enhanced transcription initiation and increased transcription burst frequency. The other type, feed-forward X upregulation mechanism involved in the post-transcriptional regulation, including enhanced RNA stability and enhanced translation via increased ribosomal binding to X transcripts. In addition, some genes could subject to the basal level of the dosage compensation via feedback or buffering systems. This basal level adjustment could also apply to any autosomal genes when they are present in aneuploidy situation. For genes, if they are not dosage sensitive, they could avoid upregulation. Furthermore, some genes, we believe they can be subject to multiple levels of the regulations, as present here, showing these orange-red bars. They could subject to the enhanced initiation as well as enhanced RNA stability. While it's very clear diverse mechanisms of X upregulation involved in the mammals' dosage compensation, not many players have been identified. One of the players involved in the X upregulation is MOF, which is a histone K16 acetyltransferase. What we found is MOF-mediated K16 acetylation contribute to the X-specific enhanced transcriptional initiation by increase the binding of the RNA polymerase II to the X-linked promoter regions. Interestingly, when we deplete MOF by RNAi knockdown, we only found a subset X-linked genes show the expression decrease. As shown here, this is a box plot for the fold changes for X or autosomal genes grouped by their expression levels from low to high into the 4 quartiles. We can see actually only X-linked genes with medium expression levels show strong decrease upon the MOF depletion. This is confirmed by the qPCR for selected genes, X-linked genes with medium or high expressions. As shown here, 3 X-linked genes with medium expression are showing 40 to 50% decrease upon the MOF knockout, upon the MOF knockdown. But there are no changes for the highly expressed X-linked genes. So far, I tell you subset of X-linked genes are upregulated differentially. So what are those upregulated genes? How much they are upregulated? This is our ongoing studies. We have continuous collaborations with Bill Noble Lab from Genome Science to identify upregulation modes and the levels for individual X-linked genes. To do that, we perform cross-species RNA-seq analysis, single-cell RNA-seq analysis, and then using the computational models to compare the X-linked genes to their ancestor ocelots. What we try to ask is, when for the ancestor genes, they present in the other species, such as in possum, chromosome 4 and chromosome 7 genes, when they now become X-linked in the eutherian mammals, such as human and mouse, are they upregulated? And how much they upregulated? Classify X-linked genes, the modern X-linked genes based on upregulation modes and the levels can help us to search for the genetic and epigenetic features associated with the upregulation mechanisms. Also help us to design the screening for the epigenetic players involved in the controlling of X upregulation. We plan to do single-cell indexed CRISPR screening for potential players from our studies as well as from the literature. Hopefully, in the near future, I can tell you more about this progress for these 2 exciting projects. So go to the second question. How is the inactive X chromosome regulated? As we know, X chromosome inactivation is mediated by one master regulator exists, which is a long non-coding RNA. X chromosome inactivation established in the early development in the female embryos. And this process can be recaptured by the in vitro differentiation of the ES cells, as shown in a model here. Upon the differentiation, exist is upregulated from one of their 2 X chromosomes. The exist RNA can then code in cis the future of the inactive X chromosome, recruit the protein complexes to remove active marks, and then also add the repressive marks such as H3K27 trimethylation. In addition, the exist also mediate the compactions of the inactive X chromosomes, which can often visualized as the bar body near the nuclear envelope, as shown here. So the question is, what is the 3D structures of the compact X chromosomes at the molecular level, and how is it mediated? So to study chromosome organization, we need a new tools. In the past decades, the Hi-C, a tool for genome-wide chromosome conformation capture, have been developed. One of such methods is developed by our collaborator Xinxian Deng from hematology, named DNS Hi-C. In short, is chromatin are fixed and digested by DNS1 instead of restriction enzyme to increase the coverage. And this digested free end then will be repaired and ligated to the bridge adapter with the bulletin label. This will be promoted for the proximity ligations. So then the ends from the two different regions will be joined together. After the cross-linking, these joint ends can be purified and for library preparation and next generation sequencing. So from these, for any given two regions, we can examine their contact frequency and build up heat map for the chromatin contacts genome-wide. So now with this tool, as well as other tools such as imaging, people now understand the 3D genome organization better at the molecular levels. As we know, DNA wrapped with histones form the 2D chromatins. These 2D chromatins can further folded into loops. These loops are majorly mediated by the cohesin complex, ring complex, which I will discuss more. And these loops actually are very important, specifically at the promoter enhancer contacts. As shown in one example here, at this promoter, using as this promoter as a bait, what you can see there's a multiple contacts, and this represented as these peaks, suggesting they could potential enhancer regions to contact these promoters. In addition, the loops are clustered together to into the isolated domains named topological associated domains. So the loops within the same domain can have more interactions than the regions between two tags. And the tags are often separated by the strong boundary elements as well as the boundary binding proteins such as CTCFs. So from the contact heat map, usually you can see this is represented by the triangle as shown here. So here is two tags. These tags usually range from 40 kb to 3 megabits. Tags will further arranged into the compartments, which is active tags, they are clustered together, and then inactive tags, they are clustered together. Then basically from a chromosome-wide view, you form two types of the compartments. One is called A compartment, usually represent euchromatin, and the other type is the B compartment, usually represent the heterochromatin. Actually, this is really consistent with classic view from the electron microscopy. What you can see as the yellow color shows here is a heterochromatin cluster. Furthermore, from single chromosomes, actually they occupy the chromosome territories. So from the Hi-C study of the genome-wide contacts, what you can see is most of the contacts is around the diagonals, and each square actually is represent a single X chromosome, suggesting most of the contacts occurs in cis from the same chromosomes. Again, this is consistent with classic DNA fish using the chromosome painting probes. As shown here, the chromosome 2 and chromosome 9 occupies a different nuclear space. There's limited interaction between them. So now we have the tools to study chromosome organizations. We also need the system to allow us to distinguish the alleles, so help us know which X chromosome is active, which X chromosome is inactive. We have two mouse models based on the cross between two mouse species. The SNP frequency between these two species allow us easily to differ the parental alleles. This class is based on the musculus black 6 females crossed to the mus spiratus males. And in addition, we also need a skewed system for the X chromosome inactivation. So since in female cells, usually they have random X chromosome inactivation. Doing this, we introduce the existing mutations or HPRT mutations into the black 6 moms. So from the F1 hybrid, we can have the F1 hybrid selected for the skew X chromosome inactivation system. As in the existing background, the X chromosome inactivation will be skewed towards the spiratus X chromosome, while in the HPRT mutation background, the cell culture derived from this mice, F1 hybrid mice, can be selected by the het medium to allow the HPRT to require HPRT expressions. Then the X chromosome inactivation will be skewed to the black 6 X chromosome. So now with these systems, we can do a lot of the allele-specific analysis, including high-seq, RNA-seq, dark-seq, and chip-seq. In addition, with the passkey cell lines derived from the F1 hybrid, we also can easily do the dosage manipulations as well as the genome editing. So we performed the allele-specific high-C in the passkey cells. Here is allele-specific contact map on the active X chromosome from the centromere and to the telomere. What you can see is this is very classic of the chromosome-wide contact heat map. Along the diagonals, most of these contacts are very heavy colored and suggesting they are present the tight structures. While chromosome-wide, we can see is there's a chessboard patterns for the chromosome-wide heat map, represents the AB compartmentalization. In contrast, when we look at the heat map on the inactive X chromosomes, first we will notice even they have the contacts along the diagonals, these triangle structures actually become weak, suggesting the tight structure become weakened. In addition, we don't see this AB compartment, the chessboard structures. Instead, we found there's two subdomains separated by a boundary. We named the subdomain 1 and subdomain 2. And within these subdomains, we see there's enhanced long-range contacts, but seems they are not so specific because we see the uniform contact, long-range contact. This difference between the active and inactive chromosomes can be more apparent when we build up the ensemble 3D models based on the contact heat map. You can see here, compared to the active X chromosome with one globule structures, we now see two globule structures, and these structures separated by the boundary regions seems with a twist. That's we name these boundary regions as the hinge region. And this bipartite structure also be observed in the in vivo tissues, such as brain. And the other groups also find this structure actually observed in the human cells, suggesting this is a conserved structure maintenance for the inactive X chromosome. So what is the function of this bipartite structure of the inactive X chromosome? And how it is this mediated? So let's zoom in for this strong boundary. What we can see is this boundary actually including at least two important elements. One is named DXZ4, is a macrosatellite repeat regions. It's enriched with more than a dozen CTCF forward motifs. We know CTCF actually can mediate the installations by forming the anchor sites for the loops with a pair of a convergent CTCF binding, which is forward versus a reverse motif. In addition, there's another mini satellite repeated regions named DSTR. Due to the mappability problem, even the qPCR shows the CTCF binding here, but we don't know their CTCF binding motif orientations. So I edited these regions by CRISP and delete the whole regions for more than 120 kb, or delete the 40 kb of DXC4 regions. In addition, I created the inversions. So now all the CTCF motifs become from the forward becomes the reverse. So what we found is, compared to the wild type, here is the Pearson correlation for the contact heat map from the inactive X chromosome. What we found is deletion of the hinge or the DXZ4 disrupt the bipartite structures. Specifically, we see the boundary disappears, and then the subdomain 2 is largely disappears, while there's less effect for the subdomain 1. Interestingly, when this region is inverted, what we see is this bipartite structure is still retained. However, the subdomain 2 do become weaker. In contrast, the subdomain 1 become even stronger, as shown here. So our models is the DXZ4 with a bank of CTCF forward motifs. They probably provide a strong platform to anchor the regions contact them, specifically anchor the regions with the reverse motifs from telomeric regions in the Y type. However, when this region is deleted, this structure becomes disrupted. In the situation of the inversions, now this DXZ4 actually provide a platform to interact from the regions from the centromeric regions. That's why enhance the subdomain 1s. So we now understand the molecular mechanisms to shape these structures. So what is their roles in the X chromosome silencing? So we performed the single cell omics studies for the ES cells during differentiation. We assayed the cells from different time points from the differentiation, day 0, day 3, day 7, day 11, as well as well-differentiated neuron precursor cells at the differentiation ends. So for individual cells, we performed the high-seq, ataxic, or RNA-seq. Then from this, we can survey do these cells already undergo X chromosome inactivation by reduced X chromosome the expression, reduced X chromosome chromatin accessibility, or form the bipartite structure. Interestingly, from this dynamics, what we see is the gene silencing as well as the loss of the chromosome accessibility on the inactive X are really correlated tightly. And this can be observed as early as day 3. You will already see 30 to 40% showing the sign of the silencing. And at the middle point, day 7, we see around 80% of the cells showing the signature of the gene silencing or lose the chromatin accessibility. In contrast, what we see is at the early time point, we don't see this formation of bipartite structures, while only at the day 7, we see about 44%, and then slowly increased. But even at day 11, we only see the 50% of cells have this bipartite structure from the inactive X chromosome. Only in the well-differentiated cells, NPCs, showing near the 80% of the bipartite structure. So this suggesting formation of the inactive X chromosome structure is probably a late event during differentiation. This is actually a time window similar to the other late events, such as later DNA replication, hypermethylation at the CPG island for the inactive X chromosome, also suggesting the bipartite structure probably is another layer for the maintenance of or the lock-in of the silencing status for the inactive X chromosome. Indeed, when we survey the expression change after the hinge deletion, we don't see the dramatic chromosome-wide reactivation from the inactive X chromosomes. We only see a dozen of X-linked genes showing the upregulation from the inactive X chromosome after hinge deletions. This is again consistent with the field. The other studies means the multi-layer of the regulations involved to mediate X chromosome inactivations. You disrupt one layer, usually will not cause the chromosome-wide reactivations. So far, I'll tell you DXZ4 play a sixth role in shaping the inactive X chromosome bipartite structures. We also find that another no-coding RNA named FIRE play a chance role in maintaining H3K27 translation, as well as nuclear positioning of the inactive X. This work recently published, and if you are interested, please check our paper. Due to the time limits, I will not elaborate this. So our ongoing and future studies following these questions is we try to examine the dynamics of X chromosome regulations in vivos, specifically in the early mouse embryo development. This project is a part of the UW4D Genome Center work, which are supported by the NIH4DN consortium. We're also interested to identify the partners interact with DXZ4 or FIRE in terms of the DNA regions, RNA, and protein. This is collaborations with David Schechtner lab from pharmacology, as well as Brian Burwell's lab from genome science. So now I tell you both active X chromosome and inactive X chromosomes are subject to the multi-layer of the regulations. So what are the impacts of X chromosome regulations on development and disease? So early studies from Brian Turner's lab shows the high expression levels of X-linked genes in female cells versus male cells in the ES cell, as well as the early differentiation times, as shown here by the bulk RNA expression studies. You can see the X2A expression ratios as a relative expression difference between males and females. You can see high expression levels for X-linked genes in females. This difference slowly disappears upon the differentiation, accompanied with the X chromosome inactivations. So, as I mentioned, we performed a single-cell RNA-seq for the different time points for the ES cell during differentiation. Our single-cell RNA-seq reviews these dynamics of X chromosome regulations and sex differences during the early development. As shown here, this is unsupervised UMAP clustering of the ES cells during differentiation based on the single-cell RNA-seq transcriptome. We can see the clustering is really nicely clustered away the day 0 cells from the differentiating cells. In addition, the early differentiate time point, such as day 3, also clustered away from the middle and the late time points, while day 7 and day 11 are more close together. While these cells be labeled by their sex, males or females, we see the undifferentiated day 0, as well as the day 3 early differentiated cell lines. We see a cluster, separate clusters between males and females, suggesting the sex differences. While for the middle time point or late time point, we don't see such a separation, suggesting this probably is because of the X chromosome regulation difference in the males and females. This is supported by the examination of the X2A ratios in the individual male and female cells. We can see the day 0 cells or the day 3 female cells. We see the X2A expression ratio is higher than 1 because of the presence of 2 active X chromosomes, while this ratio slowly go to the around 1 upon the differentiation, accompanied with the X chromosome inactivation compared to the males. Furthermore, we performed the expression trajectory analysis for male and female cells during differentiation. While they both show a very nice pseudo-time trajectory from day 0 to day 11, we do see a difference between males and females developmental timelines. Specifically, if we put the day 0 start point as 0, and the day 11 end as 1 as the pseudo-timing scales, and then we calculate the cells accumulated along these timelines, we can see a clear, significant delay of the female cell development compared to the male cells. Actually, this is consistent with early studies from Edith Hurd lab showing 2 active X chromosomes in female cells actually block differentiation. So there's an interplay of the initiation of the X chromosome inactivation and then kickoff of the differentiation. This could possibly contribute to the sex difference observed in the early embryo development in the males and the females. In addition, another fact we need to consider about the X chromosome regulations on the development and disease is escape gene. So as we know, X chromosome inactivation is not perfect. There are 3 and 15% of mouse and human genes escape from X chromosome inactivations, respectively. There are 3 major escape genes. First is the PAR genes. They are genes located on the pseudo-autosomal regions. So then these regions are present both on the X and Y chromosome. So they will produce similar product levels between males and females. The second group are the X-linked genes escape, but they also have a Y homologs, such as DDX3X, KDM5C, DDX3X of the Y homolog DDX3Y, and KDM5C also have Y homolog genes. So this also the Y homolog genes can provide the additional products and will be again this will be have a similar expression levels if you add the genes escape genes from both allele or the X-linked and Y genes in the male. The third group actually are the escape genes. They don't have Y genes, such as CAR5B and PBDC1. So this group of genes will produce a potentially higher expression levels since they express from both alleles compared to males. In addition, I want to mention, due to the expression levels on the inactive X chromosome for escape genes usually lower compared to the alleles on the active X chromosomes. Overall, these escape genes suggesting they probably fairly dosage sensitive, and their levels of the expression probably need to be tightly controlled. Indeed, these escape genes already be implicated in the phenotype observed in the X aneuploidy, such as the XXY Klinefelter syndrome and the triple X syndromes. They have the additional dosage of the escape genes. In addition, in the Turner syndrome, they actually have a reduced dose of this escape gene. So how are escape levels of this gene regulated? This is an ongoing project. We know, as I showed you before, we know the deletion of the hinge will disrupt this bipartite structure for the inactive X chromosomes, while deletion of the fire can abolish of the H3K27 trimethylation enrichment on the inactive X chromosome. And I also show you there's no chromosome-wide reactivation. That means the compromise of gene silencing from the inactive X chromosome upon the deletions of hinge or fire. However, interestingly, when we survey the escape genes in the Y type, already escaped in the Y type, we found most of these escape genes actually show significant increased expression levels upon the from the inactive X chromosome upon the deletions of fire or hinge. This really suggesting actually chromosome environment on the inactive X chromosome constrains the expression levels of this escape gene. In short, that means if we study the escape genes and any disease associated with their mutations, or the altered dosage, we need to consider about X chromosome regulations applied for these escape genes, as well as their levels. One of such escape genes we are interested is SMC1A. SMC1A escape in human, but not in the mouse. It encodes a core subunit of the cohesin. SMC1A can have self-fold and then form the heterodimer with SMC3 through their hinge domains. Then the other end, their heads can interact with other cohesin subunits to form the ring structure for hold the chromatin as well to mediate the chromatin folding. This is very important for the cis chromatid holding and segregation during cell dividing. And for the interface is very important for the loop mediation regulations, as I showed before. So we know the pathogenic SMC1A variants mostly dominant and mostly are dominant negative, count for 5% of the variants associated with one disorder named Cody-Long syndrome. Cody-Long syndrome is a multi-system developmental disorder. There are phenotypes including distinctive facial features, growth retardation, short stature, and limb abnormalities, intellectual developmental disabilities. And showing one picture of the patient here, you can see they have the distinctive facial feature as the elongated eyebrow. Interestingly, recently, a unique cohort of rare SMC1A mutations have been reported in the girls with early onset developmental epileptic encephalopathy. As we know, only 27 mutations be reported or actually discovered from our collaborators, including 4 patients from our collaborator, Xiuhua Buzaz from Children's Hospitals, and as labeled here, P1, P2, P3, P4. P2, P3 already be reported from other studies, and the P1, P4 are not reported. From the first view of the location of these mutations, we don't see a bias towards certain domains. However, when we take a close look at these mutations, we found most of these mutations actually are the mutations such as frame shift or nonsense mutations lead to the loss of functions, as well as the splicing mutations as observed in the P1. Only three missense mutations and 1 in-frame indels are present in this cohort, including the patient P2 is a deletion, and patient 3 is missense from our own patient. Interestingly, what we can see is P2 and P3 patient mutations actually located within or close the hinge domain. This domain, as I mentioned, is very important to form the heterodimer with SMC3. So it's possible these mutations affect the assembly of the cohesin complex, then decrease the number of the cohesin complex, have a similar effect as the loss of function mutations. The new phenotype caused by the SMC1 loss of function mutations is characterized by intractable early onset epilepsy, profound to severe intellectual disability, and stereotyped movements such as hand wringing. This is one picture from patient 2 at 8 months old. You can see they don't have classic facial features usually associated with Cody-Long syndrome. However, she has a very early onset of the seizure at 2 months old. Interestingly, this phenotype actually is highly assembled to the phenotype observed in the Rett syndrome caused by mutation of MECP2. MECP2 is another master regulator of the chromatin through the binding of the methylated DNA involved in the heterochromatin regulation and the gene repression. So we think maybe there's a dosage sensitive role for SMC1A in chromatin regulations, which is important for the function of the brain. considering the brain is the most complicated organs and with billions of the post-mitotic cells, neurons and non-neuron cells. So for the post-mitotic cells, any damage to the chromatin regulation could be accumulated because without the dividing, they cannot buffer in these effects. To study these mutations, we assembled a team with collaboration with Dosage from Children's Hospital, as well as Xinxian from hematology. So here is our approach. We try to, so far, we already enrolled patient 1, 2, 3 in our studies. We collect the blood cells and isolated the PBMC from the family trails. And these cells will be reprogrammed into the induced pluripotent cells. Since SMC1A escape X chromosome inactivations, and most of the patients with SMC1A mutations actually has a random X chromosome inactivations. So we can derive the patient clones with the mutations on the active X chromosome or on the inactive X chromosome. Then we can have the cells with high mutant dose or low mutant dose. We also derive the parental controls. Then we perform the 2D and 3D neuron differentiation. From the 2D differentiations, we will go for a linear differentiation into the neuronal precursor cells and further differentiate into the neuron subtypes related with the phenotypes, as well as the non-neuronal cells. For the 3D differentiation, we will go to the cortical organoids, allow us to examine the effects on the hierarchy neuronal network organization. Besides the high dose model or low dose model, we also will considering about the mosaic model, which is starting with from a mixture of the cells with a mutation on the active or inactive. This will be mimic the in vivo situations observed in the patient with random X chromosome inactivation. And these models will allow us to do the high-throughput omics or the perturbations with the linear differentiation, or evaluate the complex features from the 3D organoid models. Interestingly, from the patient 1, we already derived the neuron precursor cells. Interestingly, we see a genome-wide, even though it's not dramatic, it's minor, but there's chromosome-wide effects from the high-C analysis. Compared to the control moms, we can see a 20 to 30% decrease of chromosome numbers for all the chromosomes except X chromosomes. And this decrease is more abundant for the high dose cells, high mutant dose cells. One exception is X chromosome. While the cells with the low dose mutants also show the decreased number of the loops. In the cells with high mutant dose, that means mutations on the active X chromosome, actually we see an increase of the loop numbers. How could this happen? I already tell you, humans also have these bipartite structures. These bipartite structures, even without the allele-specific analysis, can be observed from the ensemble contact heat map from the active and inactive X chromosome, because this structure is so dominant. As shown here in the female cell line public data from IMR90, what you can see is there's bipartite structure separated by DXC4. This is different in the male lines for the active X chromosome. As shown here, this is a leukemia haploid cell lines with only active X chromosome. You don't see this bipartite structure. Then we go to the NPC cells from our patient family 1. We can see is our mom also shows these bipartite structures separated by DXC4. While in the patient cells, we can appreciate there's the disruptions of these bipartite structures, specifically in the cells with the high mutant dose. You almost don't see the boundaries. And now this structure more like have the AB compartmentalizations as the active X chromosome. This is more obvious when we check the Pearson correlations. You can see for the mom control cells, they don't have these chessboard classic AB compartmentalizations. While in the mutant cells with low dose or high dose, this chessboard structure appears specifically more effect in the high dose levels. Really support maybe the cohesion levels are very important to regulate the bipartite structure of the inactive X chromosome. While this effect is not observed in the chromosome autosomes, such as here, showing here, chromosome 7 with a similar chromosome length as chromosome X. Our expression study also supports this. So here we can first focusing on autosomal genes. We can see actually there's more genes showing downregulation consistent with the cohesion loop usually mediate the promoter and the enhancer contacts. One exception actually is X-linked genes in the high dose mode cells. We can see actually is more genes show the upregulation. That's consistent with partial reactivation from inactive X chromosome. And this is further supported by the X2 media ratios we assayed in this control or the cells with low dose or the high dose. More interestingly, we found in the high dose cells, we have the dysregulated genes enriched in the pathways important for the neuron differentiation, such as adherin, integrin, and the Wnt signal. But this actually is not observed in the cells with low dose mutations, suggesting they are very dosage sensitive. When you have more reduced cohesion, probably will have more dramatic effects. The second model is the 3D models. This is a protocol adapted from Jessica Young's lab. In short, is 4,000 cells be seeded in the individual wells to form the aggregates, and then this can be embedded in the material and transferred to the plates with swelling and pulsed with the different growth factors to stimulate their 3D structures. As shown here, this is a section of the 10-week-old brain organoid. We can see there's a complicated structure organizations. As zoom in, what we can see from the immunostaining for the SOX2 and the CYP2 counterstained by the DAPI, we can see the subvesicular zones enriched with SOX2 positive stem cells, while the deep cortical layers enriched with C2 positive neurons, suggesting there's a hierarchy networks from this organoid. This is further supported by the single-cell RNA-seq analysis for a 14-week cortical organoids. UMAP clustered for these 8,500 cells into 14 clusters, including well-known clusters, well-known cell types we know in the brain, as such as glutamatergic exciting neurons, epigenetic inhibiting neurons, NPCs, oligodendroid cells, and astrocytes. So these models will allow us to carefully examine the effects of the SMCY mutations on the different cell types and as well as on the 3D organization of the neural networks. So our preliminary study from the patient family 1 actually do show some phenotype in the growth of this or aggregation of these organoids. As shown here, this is day 31. We can see actually a reduced size, specifically in the cells with high mutant dose. So we are very excited to continue these studies and including the phenotype analysis with the immunostaining or the patch clamped electrophysiology by supported from our collaborator from Xiuhua Braz from Children's Hospital and their brain research institute. With support from the ice cream core, genomics core, we're also excited to planning to do the single cell RNA-seq as well as the spatial transcriptome analysis for these organoids. So in summary, the take-home messages is the active X chromosome is subject to the transcriptional and post-transcriptional upregulation. Subsets of X-linked genes are upregulated differentially. And the DXZ4 have a new role to shape the bipartite structure of the inactive X chromosome. X chromosome dosage regulation provide opportunities for sex difference, also impacts human diseases. Last but not least, I want to thank my lots of people. First is my senior faculty mentor, also long-term collaborator, Christine Dosage, with all the help through my junior career development. I also want to thank my current and former lab members, specifically Gala and her and Anna involved in the works I presented today. I also want to thank many of my collaborators from other departments, from UW, as well as from other institutes outside, specifically Bill Noble's lab and Jay Shendu's lab for data analysis and the support of the sequencing. And then Zhijun Duan's lab from the hematology supporting for all the high-C analysis. Yajuan Liu's lab for the cytogenetics and the genomics support. I also want to thank specifically thank the Xiuhua Buzz lab to study the new collaboration of the SMC1A, which now is we aim to for the resubmission of R1 proposals. And special thanks to the SMC1A patient families for their support for the basic science research, and as well as the funding support from the NIH. Okay, I'm now glad to take any questions.
You can type your questions in chat too if you're worried about not getting recognized or heard.
Oh, the question is say I'm not able to see the live stream in the chat, so I don't know if
yes, I will mediate the questions if anyone types one in. So Xinxian, are there any other major diseases that you think that are associated with this phenomenon in general?
Yes.
Like what are some of the prevalent ones?
So the one type of major disease I didn't talk about is aneuploidy, right? So those with the chromosome abnormal the dosage change. That's X-aneuploidy, like the XXY Klinefelter syndrome and the XO syndromes. We also have the mouse model to study that. And the other thing is for X-linked genes actually is say MECP2, the Rett syndrome. So Rett syndrome is also very interesting gene. Actually, it does not escape X chromosome inactivation. However, the patients also often have the random X chromosome inactivations. I mean, those usually also will be the heterozygous females because males usually died. But they also have them, suggesting probably at the early development, MECP2 maybe it's not required for the viability. But during the brain development, MECP2 actually is important for the regulations. But those patients have the random X chromosome inactivation. So that means some cells have the Y type, some cells have mutations. And this probably can affect this specifically in the neural network because they need to have the communications between neurons.
That's awesome.
Yeah.
Do you think that some of your work relates at all to some of the autosomal diseases that sort of show non-Mendelian patterns?
Autosomal disease, yes, that's why we're also interested in the dosage regulations of any genes if they have the aneuploidy, right? From the study from Down syndrome, actually, they also show that, the change is not really linear with the change of the gene dose. So,
wow.
So that's why the dosage compensation could be applied, or at least partial can be applied to the other aneuploidy effects. And we are also interested in if we identify epigenetic players from the X chromosome regulation models, right? Can we manipulate this epigenetic player to the autosomal aneuploidy to rescue them?
That's cool. So you're developing, thinking about or developing technology to use the X chromosome inactivation machinery to correct
or upregulation, right? Or say players involved in the escape. Can we doing this epigenome editing?
That's really cool, Xinxian. That's innovative.
Next, I have a question about the genes that escape on the inactivated X chromosome. How much variability is there in the somatic tissue? Like, is there, are some tissues have much higher escape levels versus others?
Yes.
And does it change with age?
So the mouse, they don't have many, so only have 3 to 7% of escape genes among the tissues. But humans, there's about 10 to 15 genes escape in most of tissues, but there's additional 25% cell genes actually escape variably among the tissues. So like the brain actually has the least, I believe the least escape genes, because they are, I think the model is the brain is need really tight controls of this dosage. But then like for the liver or kidney, actually, I think they have the more escape genes. Also, that's why it contribute the more high risk of the autoimmune disease in women, because they could have the effects and then they cause more escape. Actually, people already think that's why lead to the high risk of the autoimmune disease in the woman.
But does that or can that change over time? Like
Yes. Aging definitely provide because aging affects the DNA methylation, right? DNA methylation is one of the major maintenance players for the X chromosome activation. So we are also welcome to any collaboration study the aging on the X chromosome activations.
Xinxian, are there patients with mutation in SMC3 that have also this type of disease in the brain?
Yes, thanks to mention that. Actually, it's very interesting. So far for this new type, this DEE type, epileptic type, no other cohesin subunit be involved. Only one is a cohesin regulator, HDAC8, seems have one case reported have this epilepsy type. I think one explanation is, first, the other cohesin subunit probably is autosome linked. Then in this case, if it's heterozygous, the cells all have the reduced amount. Will not have this like SMC1A, have the heterogeneous populations of cells with more, with the different dosage effects. So that's a very interesting question because, and then actually the another protein cohesin is called the STAG1. No, STAG2 is X-linked, but it's not escape X chromosome inactivation. And also it has a redundant function at STAG1. So in this case, they can probably compensate themselves.
Okay.
Xinxian, can I ask a question?
Sure, yeah, go ahead.
So with respect to the structural regulation of the chromatin and how it causes or seems to be associated with the silencing, how much of that is tied to the docking of the chromosome to the periphery, kind of like heterochromatin? Is that a requirement? Does it need to be docked on the side of the nucleus, or is it?
Good question. Very good question. So we try to follow that. Like, is this needed? So the study from the Mitch Gutman's lab from Stanford actually showing the laminin association in vitro. They think the laminin association is needed for the initiation of silencing because they knock out of the lamin B receptors and they see the disruption of the initiation of the silencing.
Oh,
However,
wow.
in the in vivo study, in the mouse knockout, conditional knockout, in the mouse knockout of this lamin B receptor, actually they found no, the silencing still can be established. So
Wow.
in vivo seems they can have a way to avoid that. So it's probably another level of this mediate the X chromosome inactivation, but it's not absolutely needed.
But it helps.
So interested to follow that. Yeah, we think about if we can tether the X chromosome before this differentiation to the say the envelope or the other like the nucleolus. Do they have the effect of the X chromosome inactivation?
Wow. Yeah, you're a wealth of knowledge, Xinxian, and that's a really innovative set of studies you've conducted. Impressive work. So does anyone else have any questions for Dr. Deng? No. Are there any closing remarks you'd like to share, Xinxian?
Nope. I think that's all.
All right, well, let's everybody.
Thank you
give
guys
Dr.
very
Deng
much.
Yeah, let's give Dr. Deng a round of applause.
Thanks.
Virtually. All right, well, thanks, Dr. Deng. That was great. Scott, are there any closing remarks we need? Okay. Well, great job, Xinxian. That was awesome. I feel like I learned something. Thanks for sharing your research with us, and we will see you around.
Yeah, thank you.